In 1953, Hershey and Chase conducted an experiment to prove that DNA was the genetic material. This was the second experiment conducted to prove that. They used T2 bacteriophages to prove in this experiment, since T2 does contain 50% of protein and 50% of DNA. So what they did also is they used isotopes to prove this. They used the 32 phosphorus and 35 sulfur isotope. Since 35 sulfur contains protein and no DNA, and the 32 phosphorus does contain DNA material. A bacteriophage attaches to a cell, and it will go ahead and inject its phage into the cell. Then where the cell will replicate, the phage does replicate in DNA. So they did know that. So what they did was that they used E. coli for this, and they went ahead and used bacteriophages to affect the 32 phosphorus with 32 phosphorus, and then they used the 35 sulfur. They let these cells sit, and then they went ahead and blended these together. Once they blended that together, they went ahead and reintroduced some cells to prove this. What they did was they went ahead and used the 35 sulfur, and what they noticed was that when they went ahead and replicate these, there was no change in these at all. So they proved with that experiment that protein was not the genetic material. However, what they did notice was that when they introduced the 32 phosphorus ones, there was a change during during replication, which proved that DNA was the genetic material.